Thank you very much. Sequenceserver?
It's here.
Yeah. Okay.
Sure. 1 second.
It's not
No.
presenting. Okay. Escape. Escape. What?
I think those were in the presenter mode, that's why.
Yeah. Tools.
View. Last one.
Just ledger.
Hello everyone, I'm Anurag. So I've actually been trying to get on this stage back since 2016, and here I am actually this time. So thanks a lot for this opportunity, and thank you all for being here. So what I want to talk about actually is version 2.0 of a software that we developed. But before I go into that, a quick background about what the software is all about. So a BLAST is a really popular tool to compare sequences, and with the drop-in sequencing prices, that has created a lot of opportunities for novel research, which relies on comparing newly obtained and previously known sequences. The example use cases could be very simple, to locate a genomic region of interest in an assembly, or even to just look at the pairwise alignments visually to characterize gene functions and so on. So generally what is done is you just go to NCBI and run a BLAST search there. But a lot of new sequences, it takes time to be uploaded. And then long searches can be terminated on these servers. So what you may have to do is run BLAST locally. To do that, you can use the command line, or you can use a really nice web interface that is what Sequenceserver is about. You just give it the location of the directory containing your FASTA file sequences, and it will do everything for you, including downloading BLAST, the software itself, formatting the FASTA files into BLAST databases, and creating the search form. Very briefly, this is how the results page look. There's an alignment overview on top, a table of hits, and then you can zoom down to the pairwise alignments. I have been using my own software for when I was a research assistant. Currently, I'm doing a PhD. And it quickly became apparent to me what would be really helpful would be to have a couple of visualizations, really standard ones, to facilitate interpretation of results. So for that, we were faced with two options. The first option was to generate some static images, a PNG file on the server, and just insert that in HTML. Or try to use one of the JavaScript libraries, because the JavaScript library for visualization, the ecosystem is huge, and you can create interactive visualizations. So that is the approach we took, and for that, we export all the results, the BLAST results, to the browser, and that has enabled us to reuse some of the existing visualization libraries. So this is the example of the first visualization that we have included. The whole idea is to provide a summary of all your queries and all hits all at once. In this example, I'm just showing one query against one hit. And the green region was the query of interest, and what we wanted to see was if that region is conserved in another well-studied species, and boom, just very apparent that it is actually rearranged in this other species. The other question was, a lot of times we're working with predicted protein sequences that are not yet verified. And to be able to verify are they correct or not, one thing we often do is go to SwissProt, take a BLAST, and compare the length to the most similar hits. And this one, right away tells you, the most similar hits have a length of around 400 amino acids, but my predicted sequence falls 50 amino acids short. So clearly, there is a problem with that sequence. So further investigation, we realized there was a frame shift. This is another example. This visualization is actually called Kablam. It's from Jeff Wintersinger. We integrated it here because, again, so the top axis is my query sequence, and the bottom axis is was a new assembly that I generated, basically. And why I was interested in visualizing this was because there was a really high depth of coverage in that region. And on blasting it to the new assembly, which was done using long reads, indicated that there could be a tandem duplication going on there. In this particular case, I'm still not convinced. There could be an assembly artifact as well, and that's something I'm going to investigate. In creating this feature, We also implemented a couple other features, like, there's this, you don't just have to run BLAST through Sequenceserver. You can run BLAST anywhere on your command line on a grid engine for more power, but import the results into Sequenceserver and visualize them. So any BLAST compatible XML format is fine, and that allows us to use Diamond as well. You can also like bookmark and share these results. We also have this async job submission architecture, which basically means on large websites, there will be no timeouts. And if you wanted to, you could even integrate it with grid engines. That is something I'm going to be working on my side as well. Since I've run out of time, I'll stop here because this is pretty much about it. Currently, this is in beta. We'll be making it into stable release, few optimizations, some prettification of the visualizations. And as an outlook, it's basically this. All the results are available in browser. New visualizations can be integrated rapidly. One could do interactive filtering. And finally, being able to use large BLAST searches, that's the kind of the goal. So feel free to use it and also to contribute to this. Thank you so much.